bio, and particularly we're going to need a strong understanding of phylogeny for this. So generally when you're starting out doing biology in your science course, you're probably not actually going to start with this. You're probably going to start looking at your macromolecules, like how do proteins work, how do carbohydrates work, how does DNA is made of nucleic acids, those kinds of things. I've chosen to focus on this in this lecture because this is something that does actually become important, and it will be useful for whatever biology field people go into in terms of having that background understanding, and it's also something that people tend to struggle with more. So for people who haven't done biology in VCE who then do biology at university, there can be a bit of that struggle in terms of looking at polymers and how they work, but particularly for everyone, regardless of whether they've done bio or not, this often tends to be a topic that trips up people. That and also looking at plant life cycles can be a bit trippy. So we're focusing in on taxonomy. Again, like with other science subjects, remember to break words apart for their meaning. When you come across new terminology, it's not a bad idea to look up where it comes from, so then you can use that to build up that bank. Particularly when you're looking at a field that you want to go more into, the more familiar you can get with the root meanings behind the words and those components, that's going to help you when you come across new terminology. Sometimes you're going to be able to just intuit what it means based on those root meanings. So, as I said before in the intro, we're going to be going from all life down to mammals, describing some of these different key distinctions that we have. But first, let's talk a little bit about our terminology. So we have the idea of being monophyletic. Mono meaning one, you've got the common ancestor and all descendants of the common ancestor in that grouping. Whereas you can look at a paraphyletic group, which contains a common ancestor and some but not all descendants of that common ancestor, or a polyphyletic group, which does not contain a common ancestor of the included taxa. So when we're trying to classify things in biology, sometimes people can be a fan of, in the past we tend to look more at phylogeny, right? So you might go, oh, mammals are hairy and they produce milk. Then you might go, hang on, does that make a coconut a mammal? So what the trend has been more towards is looking at evolutionary relationships and building up these monophyletic groups. So sometimes this will be called cladistics, and we'll talk about having things in a particular clade. So those terms can be used interchangeably when we're looking at a clade and these monophyletic groups. So to start off with, what type of grouping would B and C, without including any of the common ancestors, be? So we're just looking at descendants BC, and none of those ancestors are present in the forks. Right, so remember when we're looking at any of this kind of tree, we've got some descendant here that branches off into different paths, then we've got some ancestor here, until eventually we get the extant species, the ones that exist now. Right, to give you a glimpse here, we're having different answers. I will give you a hint and say that the most popular answer is not the right one. For this question, we're just looking at these two. Again, at the moment, paraphyletic is the most popular answer. It is not correct. Right, this is why I do the practice questions on this, because this trips people up, particularly paraphyletic versus polyphyletic. Right, yeah, we're definitely getting closer here. This time it is safe to follow the crowd because polyphyletic is correct. We're not including any of the ancestors here. If it was paraphyletic, I'm just gonna... If it was monophyletic, it would, and we had B and C in there, it would need to be this whole giant group. Right, forgive the messy ink there. Because if we're including B and C, we also need to include all of their most recent common ancestors up until here. If it was paraphyletic, it might be something like, including everything there and all the ancestors, but not A. So to confirm again, the correct answer is polyphyletic. We're going to do another practice question on this. So I'm going to highlight the relevant ones here. So what type of grouping would C, D, E, the most recent common ancestor of C and D, so that's this guy here, and the most recent common ancestor of C, D, and E, so that's this one here. What would we call that grouping? So in other words, we're taking this taxa here and all of the descendants of that. Alright, again, paraphyletic is popular. It is not correct. Right, so I'm showing this so you can see, not on your own. This is a tricky subject. Paraphyletic is not the correct answer. Paraphyletic might be if, say, I was taking this whole thing except E. But in this case, I'm taking taxa and then all of the descendants together. Exactly, I can see someone switched across to the correct answer. Right, so one thing and all of its descendants, that makes it a monophyletic group. So I'm going to close this one off. We will do more on this and just head back here. So remember, a monophyletic grouping, common ancestor, and all descendants of that. So if I took this and all of the descendants, that I don't have to take everything that I can see. I just have to make sure that whatever I'm taking, it's all of the descendants of that thing. So if I wanted to, I could make another monophyletic group that's just C and D and this ancestor here. That's fine. So I'm choosing to start it from here. As long as I take all of the descendants, it's a monophyletic group. Or for example, I could take A and B and their most recent common ancestor here. And then I'm taking a monophyletic group as well because it's one thing and then all of their descendants. If, on the other hand, I decided to take D, E, this ancestor, and I didn't take C, then this would be a paraphyletic group because I'm taking this round one, and I'm taking most of the descendants, so I'm taking some of them, but I'm not taking all of them. Right, it has to be all of them to be a monophyletic group. Just all of the descendants of a particular point. So here we look at life from the kind of most zoomed out view, where we're looking at our three domains. We've got bacteria, archaea, and eukarya. Being eukaryotes, animals are in eukarya, and so we belong to this grouping. So I'm going to check your understanding. Eukarya, is it monophyletic, paraphyletic, or polyphyletic from what you see in the diagram? So with the colour coding, we can see that we're starting off at here. We've got some ancestor, and then we're taking all of these descendants of that common ancestor. Right, exactly. Alright, we're having a split opinion now. Okay, very undecided. So if I take this and I take all of the descendants, that makes it monophyletic. Right, that's what's happening here. I'm taking everything that's descended from one particular ancestor. I don't have to take the whole tree of life, just as long as whatever ancestor I start at, I take all of the descendants of that. So eukarya is a monophyletic group. If I wanted to make it paraphyletic, what I would do is I would exclude something. Right, so I might say I'm taking all of eukarya except the plants. Now, because I'm not including all of the descendants of that ancestor, now it would be a paraphyletic group. Or if I wanted to make a polyphyletic group, I might do something like taking cyanobacteria and animals and say, okay, well, I'm making a group of them. Right, so for example, cyanobacteria and plants can both photosynthesize. So if I decided to make a grouping based on photosynthetic organisms, that would be polyphyletic. Right, so I can see we've got a question here. Why is it monophyletic if we're not taking all the archaea descendants? We are. So the fact that in the diagram we can see them all in the same colour, we're taking all the animals, all the plants, all the fungi, everything, that makes it monophyletic. If that doesn't fully answer it, please feel free to ask again. When we take this eukarya group, every descendant from like this point is being included. Alright, I'm going to add in another question just to check understanding. So the question is, what grouping would ciliates and extreme halophiles be? And again, our options are monophyletic, paraphyletic, or polyphyletic. So the ciliates are over here. The extreme halophiles are here. So that something like, you remember from how Taq polymerase comes from Thermus aquaticus in the hot springs? Thermus aquaticus would be an example of our being able to withstand extreme conditions. Alright, exactly, perfect. That would be a polyphyletic grouping. Alright, let's continue. So as we go down and we get a bit more specific, one of the key distinctions we have is whether something is triploblastic. So something that's triploblastic contains 3 germ layers: endoderm, ectoderm, and mesoderm. In humans, the ectoderm develops into skin and nervous system. The mesoderm then develops into a range of things, including muscle, bone, and connective tissue. And the endoderm is that kind of inside layer, right? If you think of humans as being a pretty complicated tube, then the ectoderm is the outside of the tube, the endoderm is the inside lining of the tube, and then the mesoderm is the stuff in the middle. So from early on in our development, we have these different layers, and then they differentiate into all these things, and we get all these complex tissues. So with this, I've kind of skipped into, we're eukaryotes. Let's then zoom into different types of eukaryotes, and let's look at the ones that are triploblastic. Then, of the ones that are triploblastic, we're going to be looking at particular animals where we have bilaterians. So these are all triploblastic because they're all descendants of that group. They're all part of that, and they all have bilateral symmetry. So if we think about the cnidarians that we have just above on this phylogenetic tree, the cnidarians can include things like jellyfish, for example. Alright, so, or some kind of anemone. And if you're thinking about an anemone, it doesn't have that body plan where we've got that mirror image across that bilateral symmetry. So this is an important distinction. And you can see that there's a bunch of things that don't actually fall into this category. Then we're going to zoom in a bit more, and hopefully this makes the triploblast stuff make a bit more sense when we look at the idea of coelomates. So again, these all have to be triploblastic by definition. They're a group within the bilaterians. So we've got some things that are bilaterian, but they're not a coelomate. For example, flatworms have a bilateral symmetry, they follow that body plan, but they're acoelomates. They don't have a coelom. And if you look at these diagrams, you'll see that the coelom is this kind of gap in the tissue. And so here we've got this pseudocoelom, which isn't contained within the mesoderm, and it's just a gap between the mesoderm and the endoderm. And then we can see for our true coelomates, our eucoelomates, they have this coelom, this cavity within that mesoderm. And so it's going to be fluid filled, which can be great for things like using hydrostatic pressure to move around. Then within our coelomates, we go to the deuterostomes as part of another grouping. So you can see we're looking quite early on in development here. We've got these different layers. So there's the coelom, we've got mesoderm, there's the ectoderm, the outside endoderm, and then we have the formation of these gaps, right? So in a protostome, we have the mouth form first. In a deuterostome, it means mouth second. So the anus is formed first, and then the mouth is formed later. So humans belong in this deuterostome category. And then we get more specific again, saying humans belong to phylum Chordata. So chordates have these 4 key features at some point in their development that define them as being chordates. So there's the notochord, the dorsal hollow nerve cord, pharyngeal slits, and the postanal tail. So if we take a look at what that can look like, you can see those features here. This one might be testing your understanding a bit, but why might humans have pharyngeal slits in embryonic development, despite these not developing into gills like they do in some other chordates, like the bony fishes? Remember, in the chordate definition, you don't have to have them your whole life, but we show that we have this pharyngeal slit evolutionary history through our embryonic development. So what I want you to think about when you're trying to answer this question is our evolutionary history. So for example, right, the postanal tail, you could basically swap that in with the pharyngeal slits for this question, being like, we don't have a tail, but we are chordates, so what's up with that? Right, so we are grown in an embryonic sac that is filled with fluid. That isn't quite the answer that I was looking for here, but it is a kind of valid point to make. In terms of the importance for that for gas exchange, we don't really need the kind of developed pharyngeal slits that go into gills, like in terms of our survival in there, but it's certainly not a bad thought to have. And our kind of next zoom in relates a bit more to the fact that that embryonic sac is important. Alright, I can see someone's typing, so we're just going to give a bit of time here. Again, maybe think of the postanal tail as an example here. We don't have that as a feature that we can observe to adulthood. It is there in our embryonic development, and you'll notice that when you look at pictures of embryos, the kind of earlier you go in development, the more it seems like a whole bunch of other stuff. And as you get later and later, it becomes a bit more human specific, which can include getting rid of things that we don't need anymore. Alright, we'll be trying to answer this and continue on. You can finish up your thoughts. Alright, I will be kind of checking back and looking at that, and I'll talk while we're a little bit more about these features. So the notochord is that provides structural stability. So not everything here is a vertebrate, but it's important for that development. Yes, there we go. Pharyngeal slits are a sign of our evolutionary history. Perfect. So it's one of those indicators of evolution. I'm going to be a bit nitpicky here because we are still part of phylum Chordata, contained within the Animalia kingdom. So we are still chordates, but it's a sign of our evolution from the earlier chordates. Absolutely. So it's a holdover from our evolutionary history, and it reflects that. So we kind of make these adjustments and get rid of some of the stuff that we have earlier in embryonic development that are residual and we don't need anymore. So let's look a bit here. We've got our deuterostomes that then branches off into our chordates, which possess a notochord. And then you can see that we've got a whole bunch of different things here, and some different categories across on the left. You'll notice that most of these are aquatic. So what might some barriers be when we're looking at that transition from aquatic over to terrestrial? So you'll notice like pretty much everything to like here, and we're looking at it living in the water. And then we've got our amphibians. Before we get into our amniotes. It doesn't necessarily have to be directly related to this diagram, but you know, if you're an organism that's used to like living in the ocean, your whole evolutionary history is there, what might be a barrier to being on land? You know, just kind of basic things. Right, limbs instead of fins, right? You want to have a good way to move around. So you can see in some of those ones that are a bit more closely related, we have that move across where we are using more limbs, being able to, or if there are fins, they certainly have more structure behind them. While we wait for that typing to come up, I'm going to go across here to give you some more hints. Think about a difference between potentially amphibians and reptiles and mammals. So that could be one thing to think of. Or if you want, you can go a bit further back and go, you know, why would it be, why would we necessarily care about being vertebrates? What does that mean in terms of land? So you've got a few different options here. Just gonna options. Right, temperature fluctuations in environments, different climate on land as opposed to the ocean. And particularly we're going to be thinking that often on land, you might have things, you've certainly got more extremes, so you might have to worry about things like freezing, which you might be more likely in, going deep water or particularly cold water as used to, but heat is something that's going to be a bit hard, right? And so absolutely you're going to need significant mutations and alterations. A key thing I'll be thinking about with temperature fluctuations, if you're used to an aquatic environment, you think about when we're talking about like fish or these amphibians, right, where they're still laying their eggs in the water, right, because you need to have a way to not have them dry out. And this links to what was being said before about when we think about our fetus develops in a fluid-filled environment. So a really important adaptation that we have is the amniotic egg. And that's going to mean that we can produce young on land as opposed to in the water. So great answers there. Absolutely, there's a whole bunch of different mutations required. Some of the other ones I was kind of hinting at is that in water, you've got the water pushing up on you, so you might need a bit more structural support if you're wanting to go on land, whether that's from having, vertebrates having that vertebral column, is like endoskeleton way, or you have, like, for example, insects using that exoskeleton. So there's different ways of adapting there, but you will need something. can see the development of lungs, different air exchange, limbs as mentioned before, and now the amniotic egg. So with the amniotic egg, you can think of like a chicken egg, for example, right? With this, you've got that hard outer shell which allows air exchange while preventing drying. And so we can have this on land, it's not going to dry out, there's plenty of fluid in there. Now, many mammals do not lay eggs, but we're still part of this monophyletic group. Early mammals did lay eggs. Some mammals, monotremes, still have not lost the ability to lay eggs. And the kind of live birth that we have developed on from the amniotic egg. Alright, we've done a bit of practice on this. We've got another question here. We're now looking at, we've got our tetrapods which have 4 limbs, then we look at our amniotes. Within the amniotes we have reptiles. What kind of grouping is reptiles? And so we can see reptiles, we've got the extant reptiles, so things like currently existing turtles, tortoises, lizards, snakes, crocodiles. We've got ancestors. And yeah, I will tell you that one of the answers at the moment, yep, correct. Alright, most people are on the right track here. We can't call this a monophyletic grouping because if it was monophyletic, it would include the mammals and the birds. Right, particularly the birds are a key one to think about here. If we include some of the mammal ancestors as reptiles, we could more easily decide, you know what, even though they might seem like reptiles, we'll ignore them. But birds we would definitely have to include if we wanted to try and make reptiles a monophyletic group. So with the way reptiles is commonly understood to not include birds, it's paraphyletic. We're taking our ancestor and then we're taking descendants of that, but not all of the descendants, making it a paraphyletic grouping. And now we're on to mammals. So mammals, we've got Mammalia as a class within phylum Chordata. And above that we've got clade Amniota. Mammals have mammary glands, so this is where the term mammal has come from, and which can produce milk to feed their young. I've also got some other features in there like cool brain, where we have the neocortex, which is great for our higher order thinking. All of those other things. We